So as we continue to explore molecular biology, we're going to take a detailed look at transcription. And again, this is still just an overview, but we want to understand the basic mechanism so we can understand this process better. So again, the dogma of molecular biology is this idea that we take DNA to RNA to protein. Proteins do stuff, makes a trait. Now when we look at that in more detail, we know that we have genes as discrete hereditary units. They're the sequence of DNA that encodes for a protein. And how does that DNA encode for a protein? In codons, groups of 3 nucleotides each. Each 3 nucleotides encodes for an amino acid, right? And that genetic code that we know for every amino acid, every combination of 3 nucleotides, is universal for all life. So that first step, DNA to RNA, taking the reference book and making a photocopy, is called transcription. And included on the website is a little movie that I want you to watch now. This is to remind me, to remind you to watch that movie. It's very simple, it's very basic. But we're going to look at that. I want you to look at that. Then we're going to look at the different components of transcription, and then I want you to look at it again. And that is how we can start to get it in your head. Now let's look at the basic gene structure. Every gene is going to have a region that encodes for the protein. We call that the coding sequence. And that is the bulk of the gene, right? Now there's another part of the gene that's going to be important in what we call gene expression, and that is called the promoter. So when you express a gene, it is turned on, quote unquote. You are making RNA copies of it, and those copies are being translated into protein. So you turned a gene on. So some genes are turned on at certain times, or they're turned off, i.e. you're not making a copy of that into RNA, and you're not making the protein that gene encodes. And the promoter, the sequence of DNA, just what we call upstream of the gene, where we start to read the gene, is involved in controlling whether a gene is turned on or off. Now, at the tail end of the gene is oftentimes what's called a termination sequence to show us where the gene ends. Right? So again, that's a little bit more detail on the structure of a gene. So we call it gene expression. We express a gene, we're turning that gene on, we're making RNA and making a protein. All right? And those are terms that you're going to come across again and again. Now, if we look at this process, you can break it down into 3 major, very general steps: initiation, elongation, and termination. So initiation is simply going to be that. We're going to bind a enzyme, which is a protein in this case, called RNA polymerase. It's a protein that polymerizes an RNA molecule because it's a polymer. And it's going to use the DNA, the information in the gene, as the template. Right? So we initiate by binding to the promoter region of the DNA upstream of the gene. And then this enzyme actually opens up the double helix and can then build an RNA molecule using one of the strands of the double helix as a template through Watson-Crick base pairing. And it's literally going to move down the entire gene, building the RNA molecule, and then fall off when it gets to the termination sequence. So this now is a simplified zoom in on that RNA polymerase where it has pulled double helix apart, and we're building the RNA molecule from the template of the DNA. Watson-Crick base pairing, complementary base pairing. So not only is it important for the replication of the DNA, but for making this copy as well. And it just lends itself to this, right? It's form and function is beautiful, right? Not hard, relatively straightforward. Take a look at that movie again. All right? Now, once you've done that, think about the fact that DNA is being transcribed in the nucleus in eukaryotic cells, and that RNA is then moving out of the nucleus into the cytoplasm for translation. In a prokaryotic cell, it's all happening in the same place. Eukaryotic cell, a little bit more complicated. This goes back to our cellular anatomy. Remember that nucleus? It has those big pores on it. Those pores control traffic in and out of the nucleus. And so a lot of the traffic through those pores are RNA molecules coming out so that they can be translated into a protein.